Hi everyone, my name is Tim Stanek. I run the Journal Club on Genome Organization and Disease. Today, Kirtan and I are going to talk to you about CRISPR-Cas9 gene editing technology. I'm first going to take you through how this system was discovered, and then how researchers harnessed its power to grant us affordable and precise gene editing technology. Then Kirtan is going to show us ways in which this new technique has been used in research and in medicine, as well as the ethical implications of such a powerful gene editing tool. So let's get started. Before we begin, I'm going to ask that you watch this brief YouTube video. If you haven't already seen it, please pause this and follow the link below. Once you've finished, come back and we'll continue with a more detailed look into CRISPR-Cas9. Welcome back. I hope you enjoyed that little summary. Now let's dive in. The CRISPR-Cas system was originally discovered in bacteria, where it was characterized as an immune system. When a virus, shown here as a bacteriophage, infects a host with its DNA, Cas1 and Cas2 proteins work together to incorporate pieces of this DNA into this CRISPR array locus. Think of the array locus as a police records database filled with mugshots of previous suspects. This array is then transcribed, and the resulting RNA is processed into mature RNA. These CRISPR RNAs form complexes with the Cas9 protein, which acts like a bounty hunter holding a mugshot, always on the lookout for repeat offenders. If we zoom into the CRISPR-Cas9 complex, we can see how it's assembled and how it works. First is this CRISPR RNA, which contains the viral DNA sequence here. That's sometimes called the guide RNA. The host portion of the CRISPR RNA pairs with a tracer RNA to help it bind the Cas9 protein. CRISPR-Cas9 works by first binding the viral DNA at the protospacer adjacent motif, or PAM, just downstream of the sequence that matches the CRISPR RNA, or the guide RNA. The Cas9 enzyme itself harbors two endonuclease active sites that can cut each strand of the DNA to form a double-strand break, thus inactivating the DNA. Now, researchers realized they could create a modified CRISPR-Cas9 consisting of a single synthetic guide RNA, and this guide RNA sequence can be changed to target and cut nearly any location in any genome that you want to work with. So what happens after our CRISPR-Cas9 complex cuts our gene of choice? Here, we actually rely on the DNA repair processes already present in the cell. If we want to simply disrupt the function of a gene, cutting the DNA will lead to a random filling in of base pairs in a process known as non-homologous end joining. This random mutation of the gene often results in premature termination of the protein codon sequence, effectively knocking out the gene. However, if we want to make a more specific change to the DNA, we can include a customized donor DNA that contains our sequence of interest. The cell will then use this donor DNA as a template for repair in a process known as homology-directed recombination, or homologous recombination. This donor DNA can contain any sequence we might want to introduce into our gene of choice, perhaps a specific mutation we want to study in our research. Conversely, the donor DNA could contain a correction of a mutant sequence to restore the functionality of a particular gene. The ability to modify CRISPR-Cas9 to target any locus in the genome has enabled researchers to use it for a whole host of applications, many of which you will encounter in your individual journal clubs. As discussed earlier, we can use CRISPR-Cas9 to edit specific genes, either in specific cell types for research or in an embryo to create an entirely transgenic organism. Alternatively, we can produce packets of CRISPR-Cas9 containing virus to infect adult organisms and target specific cell types or organs. In other research, we can create a whole library of CRISPR-Cas9 complexes to knock out thousands of genes, one per cell. This produces multiple genotypes and phenotypes, which we can then isolate and characterize based on our research interests. A final innovative way to utilize CRISPR-Cas9 is to use a catalytically inactive mutant of Cas9. This mutant can bind its corresponding guide RNA sequence, but it can't cut the DNA, so it just sits there. Instead, researchers tend to fuse Cas9 to a different protein, and that way we can bring that protein to that location in the genome. And various versions of this mutant Cas9 allow us to either selectively control transcription or perhaps epigenetic modifications surrounding our gene of choice, or maybe just to visualize our gene's specific location within the nucleus. Despite the huge success of this gene editing technology and research, it isn't perfect, and there are still significant concerns we need to address when we talk about using CRISPR in human medicine. To start, the portion of the guide RNA that we change to target specific the portion of the guide RNA that we change to target specific sequences is only 20 base pairs long. This sequence might occur more than once in the genome or have closely related sequences that Cas9 can still bind and cut. So we have several tools we can use to minimize those off-target effects. Additionally, we need to be able to safely deliver CRISPR-Cas9 to the specific cells we want to edit while avoiding off-target cells. We also want to be able to fine-tune CRISPR to allow for even greater specificity and efficiency. For example, instead of using an enzyme that cuts and makes a double-stranded break, you can use 2 Cas9 variants that can only make a single-strand break and therefore increase efficiency. And lastly, using the cell's natural DNA repair mechanisms allow for the possibility that larger changes in total genome organization, such as chromosomal rearrangement, may occur. Such large structural changes could have a significant impact on the expression of multiple genes, so we have to take care to control for these events. The key to CRISPR technology lies within that initial guide RNA sequence, so controlling for specificity at this step is the most important of all. But how can we do that to minimize the risk of off-target effects? Luckily, dozens of research groups have designed easy-to-use online tools to analyze a desired gene sequence and identify potential guide RNA sequences with minimal off-target locations elsewhere in the genome. Let's try one out. So the first thing we want to do is pick a gene to target with CRISPR-Cas9. For today, we're going to immunize ourselves against COVID-19 by knocking out its binding target, ACE2. And for the sake of the exercise, we'll pretend that doing so wouldn't interfere with our overall health. An optional step in this process, depending on which guide RNA tool we use, we'll want to retrieve the DNA sequence for human ACE2 using one of several databases. But the one that we're going to use today will simply allow us to input the gene name, and it will retrieve the sequence itself. We'll enter our sequence into any of the multiple tools I listed on the earlier slide. Today we're going to use one called eCRISP. And after that, we'll select the top hits that will exert the fewest off-target effects. And then we'll order our guide RNAs for cloning. So let's go. So here we are at the eCRISP website. And the first thing we'll want to do is select our organism. Now, it's already pre-selected for Homo sapiens, but if we were working with another model organism, you'll see you can choose from several different genomes. And the purpose of this, of course, is to provide the tool with the entire genome sequence to look for off-target sequences. In step 2, we want to select our target region either by gene symbol or input sequence. Like I said before, we could retrieve the sequence and paste it in ourselves, but it's just as easy here to input our gene symbol, which is ACE2. And finally, before we start looking for guide RNAs, we want to alter the stringency of our search. Because we're looking for the most specific guide RNAs with the fewest off-target effects, we're going to select the strict stringency parameter, and then we'll start our guide RNA search. And here we have our summary and our results. So you can see a total number of 778 possible guide RNAs exist within the ACE2 gene. But the number of successfully designed guide RNAs is only 14. That's fairly typical when you consider all the constraints we put on the guide RNA design, especially because we chose the strict parameter. Down here, as an example, you can see all of our different guide RNA sequences, and it shows that they're upstream of that PAM sequence that's required for Cas9 to bind and cleave. This SAE score is a predictor of how specific and efficiently your CRISPR-Cas9 will cut just this sequence within the ACE2 gene. And over here you can see that throughout the whole genome, this sequence was found with a perfect match only once. If we scroll down to the bottom here, there's a visual representation of the ACE2 gene, which, again, because DNA is double-stranded, genes can be on both strands, either forward or reverse. ACE2 is on the reverse strand. And you can see down here every single guide RNA that eCRISP tool has generated and where precisely it targets the ACE2 gene. Now, if you want full knockout of a gene, earlier is generally better. You're likely to introduce a truncation here, and you get a non-functional protein, in essence knocking out the gene. So for our exercise, We would probably choose these first 3 guide RNA sequences to test, as they're farthest upstream and most likely to result in a full knockout. Now that we have our specific guide RNA sequences, we can clone them into DNA plasmids, transfect those plasmids into cultured cells to produce CRISPR-encoding virus, harvest and purify the virus, infect ourselves with the virus, and then confirm mutation or knockout of ACE2 by sequencing a biopsy of ourselves. As a functional readout, of course, at the end of all this, we would want to test our immunity to COVID-19. Now that you've learned the details behind CRISPR-Cas9, go back and watch that video again and see what parts and processes you can identify. This wraps up my section of the techniques seminar, so I'm going to hand this off to Kirthana. Thank you.
Hey guys, I'm Kirthana. So far you guys have talked a little bit about the history of CRISPR, how CRISPR actually works, and how you can use CRISPR in your own experiments. But now we're going to talk a little bit about how to interpret other people's data. When people use CRISPR in their experiments and they publish it in the literature, in journals, you want to be able to understand what they're doing and make sure their data is valid. So we're going to talk a little bit about that. We're going to explore some of the therapeutic applications of CRISPR, and we're also going to touch on some of the ethical implications that come with this genome editing technology.
Now, as Tim mentioned, we can use CRISPR to knock out a gene. You might be wondering, why would you want to knock out a gene? What's the point of that? Well, knocking out a gene is a simple way to study the function of a gene, to see what a gene could be involved in. For example, if we wanted to ask the question, does gene X contribute to tumor formation? The way we'd address that is we would CRISPR out the gene and then perform the appropriate functional assays to determine if there are differences between the wild type, meaning the normal version of this gene, and the knockout. Now we're going to talk about some things you might encounter in the literature when reading a CRISPR paper. Generally, there are some schematics that you might run into. So here's an example of a paper that has a vector that's drawn out that contains the CRISPR constructs. So in this particular paper, there are 2 different guide RNAs that were used, as shown here. And then the Cas9 protein is encoded in the same vector as shown here. The arrows just represent the different promoters. Other instances can show schematics where the authors want to show where the different guide RNAs bind that they've designed. Usually, people design several different guide RNAs because one guide RNA might not give a good cutting efficiency. You might not get the results you want. It might not do a good job of cutting, or it might cut something completely else, or something completely different than what you had wanted it to cut. And so, generally, many guides are used and tested and evaluated. And here's just a schematic of where different guide RNAs that were designed bind to the gene of interest, or the particular exon of interest. So once the whole CRISPR process has been carried out in the way that Tim described, how can we actually validate that the genome has been edited? The surveyor assay is a quick and easy way to confirm editing. It begins by taking the genomic DNA from a cell population, as shown here, and this genomic DNA hopefully should have the CRISPR edit as marked by the red X. Then the DNA is PCR amplified, so you get a lot more of it. And then the DNA undergoes a denaturing and reannealing reaction, meaning that the DNA strands are essentially allowed to break apart, and then they are allowed to rehybridize randomly, meaning that strands could form mismatches, as shown by the bottom 2 strands here. These are called heteroduplexes, and the mismatches occur if there is a genetic edit that was successful. Then, something called T7 endonuclease is used to perform a digest. Now, T7 endonuclease is an enzyme that will create a double-stranded break at the site of a mismatch. So if you get successful editing, and then you get these heteroduplexes that are formed with the mismatches, then this endonuclease will come and cut right at the site of the mismatch. If the gene has not been edited, then there will be no restriction digest or no efficient no breaking of this DNA construct, and the endonuclease will have nothing to cut. And then to assess the success of this, you run it out on a gel using gel electrophoresis. And this is just an example where the unedited DNA, since it's a larger construct, will not migrate very far. It'll stay at the top of the gel. Meanwhile, the edited construct that has been cut into 2 pieces should generally form a couple of smaller bands because they're smaller in size. So here's an example that you can see taken from that same paper that had the schematic with the 2 guide RNAs in 1 vector. So what we're looking at here is basically the authors used individual guide RNAs and then used these individual guide RNAs in 2 separate vectors together, and then used 1 vector containing the 2 guide RNAs to see which one of these combinations gave the best editing efficiency. The nuc stands for the endonuclease, so it was either added or it wasn't, as indicated by the plus or the minus sign. And then what we can see here is that there did appear to be some successful editing that has occurred, as shown by the appearance of these 2 smaller bands. Usually, the darker the bands are for these smaller bands, the more successful or more efficient the editing was. That means you have more of that edited product. Now, the surveyor assay is great to confirm that something has been edited. But next you want to confirm that you're actually editing out the gene of interest. So to do this, another simple PCR reaction is performed. What you can do is have PCR primers that will amplify the region that you're trying to get rid of. If the region of interest is deleted, then that piece would essentially be floating around by itself and would end up being a small fragment of DNA that is amplified, as shown by these small bands right here. Meanwhile, if it is not successfully excised, then when the PCR reaction occurs, you won't see any product, as indicated by the empty lanes here. So performing a PCR is definitely important to validate the genomic deletion, but more important than that is sequencing. So generally, samples are sent out for either Sanger sequencing or next generation sequencing to confirm that you have successfully altered the gene and deleted exactly what you wanted to delete. Most papers don't usually show the sequencing data, but this is an example of something you might see where there's a wild type sequence and then several clones are sequenced, and you can see where there are mismatches or where there have been nucleotides that have been removed. You might be wondering what's the point in doing surveyor assay and the PCR prior to sequencing, but sequencing tends to run on the more expensive side, so it's definitely wise to perform a PCR and a surveyor assay just to confirm that your samples do have an edit of some sort and it seems to be pointing in a promising direction before you invest lots of money in the sequencing methods. So it's also possible to validate the knockout by looking at other aspects such as mRNA and protein. So the central dogma of biology is you have your DNA, and remember the DNA is where the genomic deletion actually occurs. This is what the CRISPR targets. The DNA undergoes transcription to create mRNA, and then the mRNA will undergo translation to create protein. So sometimes in papers, they might skip showing all the PCR data and might show you a qPCR. So this is an example where a CRISPR was used to knock out the gene p53. And p53 is a really important protein. in the context of cancer and turns on lots of different downstream target genes, such as this gene SAT1. And so here they're seeing if they knock out p53 using a CRISPR, do they still see expression of downstream targets like SAT1? Nutlin is a reagent that's used to activate p53. And so essentially if p53 is present, then the level of the downstream target gene expression will also go up. Now you'll notice there's a control CRISPR that's used here. And so what that essentially means is that this is a construct that has Cas9. It has a random guide RNA. So in theory, it shouldn't be targeting anything in particular. And control CRISPRs are important to use, or controls in any experiment for that matter, are important to use just to ensure specificity, just to ensure that whatever results we're seeing with this p53 CRISPR are not due to off-target effects. And so this, again, is a qPCR showing us the mRNA levels of a p53 target gene. We can see that when you knock out p53, this gene is not turned on. Whereas when the control CRISPR is used, it's turned on quite nicely. So it again tells you that you have efficiently knocked out p53 on the mRNA level. And to confirm that on the protein level, you can run a Western blot. And again, here is the same control CRISPR that's used. Here is the p53 CRISPR. And you can see that the bands for p53 are gone, meaning that the CRISPR has successfully eliminated the production of this protein. Now, all these proteins right here, p21, TIGR, and MDM2, are downstream target genes of p53. And we can see that when you knock out p53, the expression of the downstream target genes is also turned off, telling you that this CRISPR knockout has in fact been successful. Again, when you look at the control CRISPR, you can see p53 is there and very happy, and all the downstream target genes are turned on. Actin, in case you didn't know, is just used as a loading control to ensure that you did in fact load some sort of protein in your sample. And so now that we've talked about how genomic deletions can work with CRISPR knockout and what you might encounter when reading a paper, we want to ask the question, well, how can we harness the therapeutic potential of CRISPR? Why is CRISPR so revolutionary, and why is it such a big deal? Now, there are a lot of diseases that are driven through point mutations, meaning a simple swap in the DNA that can disrupt normal processes. Oftentimes, these are genetic diseases that are inherited, such as cystic fibrosis, which occurs where there's a mutation that occurs in the CFTR gene, or heart disease has specific genes that are altered that can be manipulated to treat heart disease, blindness, and the list goes on. Muscular dystrophy, Tay-Sachs disease, cancer. So, one way to fix this is by performing a CRISPR knock-in. A knock-in is a simple way to essentially re-engineer the genome. It allows you to remove the mutation and correct it with the specific change that you want. For example, if you want to ask the question, how can I create mutation Y by converting this A to a T? All you would do is CRISPR in the gene and then perform the appropriate functional assays, just like you did with the CRISPR knockout, to determine if there are differences between wild type and the knock-in. Now, I would like to put in a word of caution that a CRISPR knock-in is very difficult to do. The efficiency is much lower than a CRISPR knockout. And that's because this involves the use of a separate donor plasmid and requires another process called homology-directed repair. So the probability of this occurring in the way that you want it to occur becomes much lower. But that being said, it's still possible. The workflow is very similar to a CRISPR knockout, but like I said, in addition to the guide RNA and the Cas9, as shown by this second schematic right here, it requires a donor plasmid. And the donor plasmid should have the corrected form of the gene that you want to incorporate. And in addition to that, it can also have a reporter gene, like a fluorescent protein, such as GFP. That way, when this construct is incorporated into the cell, If the donor plasmid has both a reporter gene and your construct of interest, or your gene edit of interest, you can easily see if it has been incorporated by looking under a microscope and just looking to see how many colored cells there are. And that will tell you if this knock-in has been successful. So as I mentioned, the process itself is fairly similar to a CRISPR knockout, where you perform the whole transduction or transfection to introduce the CRISPR constructs into the cell, perform clonal isolation, expand them, and then validate using genotyping, PCR, Western blot, qRT-PCR, other functional assays. And there could be further downstream analysis using single molecule imaging. Now, here we're going to show a PCR that's used to confirm the knock-in. In this case, there are two primers that have been designed targeting the 5' end of the gene of interest, and then the second primer targets the site within the gene of interest. So here we're knocking in RFP, or red fluorescent protein, and the primer should bind directly to that gene of interest. So if the gene knock-in was successful, then both these primers can bind and amplify something, and you'll see a band when it's run on a gel. But if the editing was not successful, if you have not successfully incorporated this construct, then the primer too can't bind to anything. So there will be no PCR product, and that will result in an empty lane. As I mentioned, you can also use a reporter gene to verify successful knock-in. So here's just an example where a fluorescence microscope was used to image cells that have undergone a knock-in procedure. And you can see that when they're subjected to fluorescence microscopy, the red cells light up, showing that this construct has successfully been incorporated into the genome. Now, an example that's gained a lot of attention in the media is the success of using CRISPR to treat sickle cell disease. So sickle cell disease is caused by a genetic mutation that occurs in the beta globin gene. Essentially what happens is this mutation will result in a defective form of hemoglobin, and that leads to the formation of these sickle-shaped red blood cells, which leads to several other health issues and a lot of pain and discomfort for the patients carrying this mutation. A simple way to fix that is just by performing a CRISPR knock-in, where you convert the A to a T in that beta globin gene. So to do that, cells are taken from the patient and CRISPR Cas9 editing is performed on the beta globin gene. These cells are the cells are originally removed from the bone marrow. They're edited with CRISPR, and once they've been edited so that they can produce their own hemoglobin, they're reinserted back into the bone marrow. And this hopefully should result in healthy red blood cells. I would highly recommend checking out an article in NPR which talks about the success of this. This is a very new treatment. It's revolutionary in terms of using CRISPR in human patients, and the results have been really, really effective. Now, with great power comes great responsibility. And CRISPR is an extremely powerful tool, but there's still a lot that has to be learned about it. It can be really helpful when targeting diseases that affect somatic cells. For example, taking cells out of a patient's body, editing them, and putting them back in. But actually altering the human germline is a completely different story. We don't know what the off-target effects could be. We don't know how this will affect people who are CRISPR edited and how this could affect evolution and the long-term implications of what this means for the human race as a whole if we actually start editing the genome. And so a huge scandal that erupted in 2018 was when this scientist, He Jiankui, CRISPR'd out the CCR5 gene in human embryos. His goal, he had good intentions. His goal was to try and make these children immune from developing HIV because they had an HIV-positive father. This resulted in the birth of twin girls, and they appeared to be very healthy. However, this was a huge red flag in the CRISPR community. Again, just because he is the first person to have performed CRISPR editing in a human embryo. Like I said, when you actually edit a human embryo, when you actually edit the germline, the implications of what that could mean for these children down the road are still unknown. No one really knows what could happen to these kids and what could happen to their offspring in the future. So this raises a lot of ethical questions. First of all, what could go wrong with genome editing? We talked a lot about the off-target effects that could occur. There are a lot of things that are still unknown about CRISPR. So it's still pretty dangerous to perform genome engineering on human embryos. And then coming back to more of a moral dilemma, when is gene editing okay? For example, maybe it's okay to use gene editing to edit sickle cell disease mutations or cystic fibrosis. But what about things like deafness or dwarfism or ADD? Who decides what's okay to get fixed and what's not? What's a mutation that is considered beneficial versus a mutation that's considered harmful? And then also, who decides what should get fixed? Like I just said, it's a very gray area. And it can easily turn into an issue where the rich and powerful will have the ability to customize their kids, design CRISPR babies with beautiful eyes and high IQs, whereas people who have less money might not have the ability to do that. And then that begs the question, well, how will this affect the evolution of the human race? So clearly, there's a lot of work to be done in the field of CRISPR, both at the bench, at the bedside, and in terms of the ethical and legal implications of CRISPR. But it's a really powerful technology with a lot of growth and a lot of potential to make a positive impact in the world. So I'll leave you with that. Thank you for listening, and if you have any questions, feel free to email Tim or myself.